What you've heard on the afternoon program so far is old stuff. What we're going to hear on the rest of the afternoon is new stuff, and by experts in the field. We like to call Dr. Rubel the godfather of hair cell regeneration. He denies that, of course, but we still call him that anyway. And he's going to tell us how we got here and where our things are going in the short term. So, Ed, if you want to take the podium. There we are. Do you have a pointer?
Yep.
The top thing on that is a pointer.
Right. Okay. Ladies and gentlemen, thank you for coming. I hope you're enjoying the presentations and the discussions. I'd like to thank George Gates, and I'd like to thank Sherry and Andrea and all the people that have worked so hard to put this presentation or this afternoon and evening together. And what I'm going to do is talk a little bit about what I call the new era of hearing rehabilitation. And I'll tell you where I'm going. I'm going to talk about birds. Whoa. I'm going to talk about birds and hair cell regeneration in birds. I'm going to talk a little bit about fish and hair cell regeneration in parts of fish. And then I'm going to talk about what we don't know about mammals and what we do know about mammals and what are the challenges for the hearing restoration project. So let's begin. I'll begin, and I'm sorry there's a lot of overlap with some of the pictures Dr. Gates showed and were shown in the last presentation. But here's, I'm going to have trouble with this. Here's the outer ear, middle ear, and inner ear. And if we go on and look down on the inner ear, we see this beautiful picture with some fancy stains. And here we have, in this animal, this would be like a human inner ear, and we can see it spiraling down, and the green are the hair cells, and the red are the nerve fibers that are going to go and connect to the brain, and they're going to innervate the hair cells like that. And so here you see a row of inner hair cells and 3 rows of outer hair cells, and one little unit, if you will, is recreated 3,500 times in the human ear, so that one unit, if I talk about it, is 1 inner hair cell and 3 outer hair cells. Now, if we look down across the organ of Corti, we see this that you've seen before, an inner hair cell, 3 outer hair cells, bunch of support cells, the nerve fiber coming from the inner hair cells toward the brain, and other nerve fibers coming from the brain to innervate or to talk to the outer hair cells. And of course, this is what wiggles up and down that causes the stereocilia here, or these little hairs that stick out of these cells, to wiggle and sets up electrical pulses that go to the brain. If we look down on that cochlea again, in this image, the scanning electron microscope image, what we see now is the spiral down, and as Dr. Gates showed and other people showed, the 3 rows of inner hair cells. Each one of these is the top of 1 cell, each one of these, and this would be the top of 1 inner hair cell. So when I talk about a unit, here's a unit that's recreated 3,500 times across the cochlea. And if we have a fairly severe hearing loss, we might see something like this, with the outer hair cells gone and the inner hair cells remaining. And this will give you a significant hearing loss, but not a total hearing loss. But if with more damage, you'll see something like this, and all the hair cells gone and no hearing whatsoever from that part of the cochlea. Now, we talked about the number of people, and here are some statistics on the number of people in the U.S. that are hearing impaired to the point that it affects language comprehension. And We can calculate from that something on the order of 600 million people in the world. And I may be off by 1 0 either way, but it's a lot of people, okay? And if we could stop that from happening, or if we could correct it totally, it would be a wonderful thing for the world. It's not just hearing that we lose. This, by the way, says maybe it's your inner ear. The same kinds of hair cells that are in the hearing parts of the inner ear are in the balance parts, or the vestibular parts, and there are probably as many people that have balance problems due to this, and we know we get balance problems due to this with aging, as people with hearing problems. So, one of the axioms in hearing research from the time I started in it 40 years ago was when the receptor cells of the inner ear are lost due to mutations, disease, injury, or environmental toxin, there is no replacement. What you got as an early embryo, as a fetus, is all you're going to get for the rest of your life, okay? And there is a complex but direct relationship between hair cell loss and hearing loss, and I'll add a direct relationship between the loss of hair cells in the balance organs and the amount of balance problems one is going to have. That was an axiom for most of life, and what we did to correct that, and what we do to correct that in the 20th century, as we heard before, is fundamentally, at least at the level of the inner ear, there are two types of treatments. One is hearing aids for people with mild to moderate hearing loss, and the second is cochlear implants for people with more severe hearing loss. Well, what will happen in the 21st century? These, by the way, don't correct the problem, they bypass the problem. The hearing aid trying to stimulate the surrounding hair cells that are still there, and the cochlear implant directly stimulating the nerve because the hair cells are gone. Well, what will we do in the 21st century? In the 21st century, the habilitation of hearing will include hair cell regeneration, the prevention of hair cell loss due to chemical toxicants like gram-negative antibiotics and others, due to noise exposure, and the giant gorilla due to aging. We will also have gene therapy. And those are the outlook for the 21st century that we're just beginning. So what happens with this? What is our outlook? Well, before 1985, we thought of birds as having beautiful colors, beautiful song. We loved watching them in our backyards and so forth. And they're just a joy to all of us, particularly those of us who can hear. But that's the way we thought of birds, and we can't even see an ear. There's an ear flap here, but we can't even see an ear. We knew birds heard well because they sing to each other, and they must hear what they sing. Well, that was what we thought of birds. We knew that there were some birds with great lips, but not very many, that was good. But in 1987 and 88, what we found is that some birds actually have great ears, okay? Now, obviously, that's not a real bird, okay? That's a bird we made up in Seattle, but it shows the problem or the solution pretty well. What was discovered in 1987 and 1988 is quite simple, and this was one of the experiments that was done. This happened to be done in my laboratory, then at Virginia. What we were doing, as I said before, is we're looking at the cochlea. We're looking at cross-sections of the bird cochlea, and there's the bird cochlea shown here, and here is the high-frequency end, and here's the low-frequency end. And when we treated the animals with a ototoxic drug, with a drug that we know kills hair cells in humans and other mammals, we lost the hair cells. They went down almost to zero here, and even at this place, it went down very, very much. But as we waited longer, 8 days and then 22 days, the hair cells came back. And that was a huge surprise, as I said earlier. Why did those hair cells come back? That went against everything we knew in biology about ears. At the same time, Doug Cotant, shown here, who was going to be here today but couldn't make it, was doing some studies on noise damage in chicks as well, and he was looking at these cochleas in the electron microscope. And what he saw about 12 days or 8 days after noise damage was these little hair cells that I'll show you in a minute, okay, these little ones with these little, what we call microvilli sticking up, which a mature hair cell doesn't have. Doug had done some wonderful studies on development in which he found that embryonic hair cells also had these microvilli. And so he suggested, as did we, that hair cells were actually regenerating after this noise. Our papers were published about the same time. And what we see is something like this. Here's the normal bird cochlea, and you can hardly see, but this is a bunch of little spots on there, and each spot is a hair cell, and there are about 15,000 hair cells in this cochlea. And here it is magnified so you can see each of these little spots is the top of a hair cell. And here it is that same area after we expose the animal to noise. And you can see it looks just terrible, and all these hair cells are blebbing out and being discarded by the cochlea and are going to leave a horrible loss of hair cells and a loss of hearing. But if we look just a few days later, just 5 days later, here are these little cells budding up, these little baby cells budding up, surrounded by surviving cells, okay? And that's why we call them baby cells, and some of them have these little microvilli there. If we look a couple days later and do a high-power scanning electron microscope picture, we see this, with these little microvilli, the hair bundle, okay, surrounded by mature hair cells that weren't killed. Well, so we knew that hair cells were regenerating somehow, and 30 days later, looks like this. Okay, all the hair cells are back. Now, it's not perfect. There are little abnormalities like in here and stuff like that, but it's pretty good for government work, okay? It's real good, and the animal can hear. So what do we know about birds? Okay, what we found out about birds in the next 10 to 15 years, okay, I guess we call it 18 years there, is that the new hair cells, or that hair cells do regenerate, and it's not age dependent. It'll happen we actually have one colony of senile birds. These are senile quail that my colleague Brenda Riles studied, and they regenerate hair cells just as well as a baby chicken does. New hair cells are produced in response to damage in the avian auditory epithelium. It's called the basilar papilla, but that's not important. You can call it the cochlea. And new hair cells, as somebody mentioned earlier, are continually replaced in the balanced parts of the inner ear, what we call the vestibular epithelium, and they turn over at a low rate normally. That rate is increased tremendously after damage, and they restore the normal number, and then they come back to turning over at a low rate. Okay, in the auditory parts of the ear, there's no turnover. The cells are totally quiescent until we do damage. Then there's a surge of regeneration, and then it's recalibrated back to no turnover at all and no cell death. The new hair cells are appropriately connected to the brain. Okay, that is, it does no good to have new hair cells if they aren't connected to the brain, right? The ear can hear, but the brain can't hear. And they do get connected to the brain, and they're the right size and shape and number as they were in the mature animal. The new hair cells restore normal hearing, normal structure of the epithelium, and they restore near normal hearing and perfectly normal vestibular reflexes, as best we can tell. The new hair cells restore perception and production of complex vocalizations. Birds lose their song when they lose their hearing, but they gain their song back when they restore their hearing. And we found that there were a number of growth factors that were identified that modulate this progenitor population, this population of cells that's making new hair cells. It can stimulate it or it can inhibit in birds. And we found some kinds of proteins, transcription factors, as Dr. Groves explained that will modulate hair cell production in major ways. Now, when I say we, let me stop for a moment. When I say we, I don't mean me, okay, and I don't even mean the people in my lab. I'm talking about the field. And in this talk, I'm not going to bother you with a bunch of references and so forth. There are review articles. And so for the most part, I won't be mentioning all the people that did this wonderful work. But be it known that it's not all from me. Little of it is from me. Well, what's this? Well, this is a baby fish. This is a baby zebrafish. It's a larva. It's a free-swimming larva, 5 days old, and it's live when we take this picture. And what we can do is, in a live animal, on the surface of all fish, all aquatic vertebrates, as a matter of fact, we can see that there are little bunches of hair cells. This is called the lateral line system, and each of these bunches is called a neuromass. Okay, if we look down on this neuromass, okay, we'll see something like this. Okay, here's a normal neuromass. Okay, with these would be the little hairs coming out. This is a nuclear stain, so we know it's a nuclear stain just for the hair cells. And then we have this rosette, and each of these little yellow spots is a rosette of hair cells that function very much like the hair cells in our inner ear. Okay, when we treat this animal with the same kinds of drugs that kill the hair cells in us and in chickens, we lose the hair cells. Okay, if we wait in this animal just 72 hours, boom, they're all back. 72 hours, it regenerated all the hair cells. Quite remarkable, okay? What did we find? Well, what we found is not only do chicks regenerate hair cells, okay, but fish throughout their life regenerate hair cells. Reptiles regenerate their hair cells. Frogs regenerate their hair cells. So all of these animals regenerate either the hair cells on their body or the hair cells in their inner ear. So we have to ask, okay, if birds and fish and other animals can regenerate hair cells, why can't we? Why can't me? Okay, I'm getting to that age where I'd like to have those hair cells back, aren't I? And several people in the audience are getting at that age. Maybe all of you, okay? At least you can envision getting to that age. Well, in order to tell you why we don't do it, what I have to do is tell you a little bit about how it happens. Here's how it happens. We actually have two types of regeneration. One we call mitotic, and one we call direct transdifferentiation, or conversion. In the mitotic state, when we kill a hair cell, okay, and the hair cells die in a variety of ways, cells that are called support cells, these yellow cells here, leave their static state and start entering what's called the cell cycle. They go through the stages of a dividing cell. And they go through these stages, and the cell migrates to what we call the luminal surface up here. It divides, and some of the daughter cells become hair cells, and they become the exact replica of the hair cells that were at that place. In the conversion case, the damage also causes the cells to be lost. The support cells remain, and then some of those support cells kind of magically change their stripes, so to speak, and they become hair cells without going through cell division. Okay? So there are two ways that non-mammalian vertebrates regenerate hair cells. Mitotic, that is making new cells and recovering that way, or conversion of a support cell into a hair cell. What happens in mammals? Well, what happens in mammals is it stops right there. Normally, in mature mammals, the cells don't enter the cell cycle. They may go slightly into it, but they never divide and make a new cell, okay? And they never convert. And these support cells just stay support cells throughout the rest of the animal's life. Okay, so that's what happens. So the goal of the field, of course, is to stimulate regeneration in mammals. And I'm going to tell you a little bit about what's happened. I'm not going to tell you very much about the experiments. I'll give you a couple of examples of the experiments, but my colleagues who are going to tell you about their great research and more current research will tell you more about their experiments. Well, First of all, we had to develop what we call in vitro preparations. At the time that we discovered this, there weren't any cell culture preparations for mature inner ear tissue. We didn't have them in the field. So what happened is, I'll tell you, we developed them. Okay, we found that some growth factors can stimulate a small amount of cell division in the neonatal and mature mammalian inner ear and inner ear sensory organs. We also found that after damage, a small amount of proliferation in mammals, in the mammalar vestibular organs occurs both in vivo and in vitro. In vivo means in the living form, and in vitro means in a dish. So we could take out this tissue and put it in a dish. And in the mammal, in the vestibular epithelium, some division took place. That was really an exciting finding because we thought it was totally null, totally asleep, if you will. Also, we found that manipulations of some of the cell cycle genes, some of the genes that are involved in that process of cell division that had been discovered in the cancer research, some of those genes, manipulating them could facilitate cell division in the neonatal hearing and balance organs. In the neonates, in other words, even after they were born in mice, okay, we could get a good bit of regeneration or of cell division, if you will, and Andy will tell you some about cell differentiation in these cases. And we found some of these transcription factors would stimulate an overproduction of hair cells, particularly in neonatal animals. Let me show you a couple of these examples. Here's an example of work done by Liz Osterly and Mark Warshall at two different institutions. And what I want to point out here is that how basic this research was, what a difference the field was then and now. As I said, they had to develop these in vitro preparations to test different growth factors and so forth. And they put different growth factors in there and found some really surprising and really encouraging results. Some of those are shown here. I'm just going to skip over the actual growth factors. But the limitations were that mostly it helped in young animals, and there were very, very few hair cells. But it was proof of concept. It showed that there's possibility there. Okay, another approach was to manipulate cell cycle genes. Now, this shows you some of the genes that are involved in the cell cycle and in forcing cells and in stopping cells from going in. This R point is called a restriction point, and there are certain genes that are involved in stopping the cells so they don't go through the cell cycle, i.e. so they don't make a tumor, okay? And what we learned to do over this time was to take out genes in mice, and one particular gene that was taken out was this gene called P27KIP1 that was taken out by two different labs, and I'll show you the results from our lab, okay, in young mice. Okay, here's a normal mice. Here's a normal mouse. We're looking down on the sensory epithelium. Each of these spots is a hair cell, and underneath each of these spots is a support cell. And as you can see by the lack of black dots, this is an 8-day-old 8-day postnatal mouse, there's no proliferation going on. There are no black dots there, and the black dots indicate that the cells are going through the dividing process. But when we get rid of the P27KIP1 gene, we see even in an 8-day-old mouse, lots of cell division going on, as indicated by the black dots. Now, this has been done in several laboratories now, but this goes away. It goes away very fast after birth. So let's talk in the end about some of the challenges, okay? Well, successful starts we need much, much more work on the successful starts. We need to look at combinations of approaches, growth factors, gene therapy, and stem cell transplantation. These are being implemented, studies that are doing these combinations. We need to introduce stem cells into the mouse. Some introduction has been done with very, very promising results. We need to replicate and extend those results to mature animals, as I'll mention. We need to look further at the induction, what happens when we introduce the MATH1 gene. What is the cascade of events that happens in the young animal and doesn't seem to happen in more mature tissue? Okay, we need to switch our attention to the support cells. Okay, here's the cochlea that I showed you before, and here are all the hair cells. Most of auditory biology has dealt with the hair cells, not the support cells. And here are the different kinds, many different kinds of of support cells, and we need to know what's happening with each of those cells. We need to define the biology of those cells. We need to understand what happens to each of those supporting cells after different types of pathologies. In animals that do regenerate hair cells, which cells are induced to make the new hair cells, and what is their pattern? I have a bunch more challenges that I'm going to not go through them all, but there are many more challenges. We need to understand the biomarkers for regeneration. What's similar in frogs and fish and birds and not happening in mammals? What's happening in mammals that shouldn't be happening to stop the process, and what's happening in those other animals? We need to understand the epithelium of the different human pathologies. What happens to the cells under different pathologies, and we need to reprogram them. And we have to understand what's happening when the cells have been silenced. We need to understand this developmental difference in competence. We will hear more about this developmental difference in competence, and we need to understand what's different in the young animal and the mature animal. And we need to develop delivery mechanisms and methods. So the challenge of accelerating regeneration, this is going to require team research, coordinated, collaborative research on a multi-center effort. It's going to require more investigators coming from different fields, focusing on the problems of hair cell regeneration. We're going to have the freedom to explore new ideas and use new tools as they're developed in biology to maximum advantage, and we're going to have stable funding. The Hearing Restoration Program of the Hearing Health Foundation can provide this. It started, it is spearheading financial support and administrative management by the Hearing Health Foundation. The HRP is a new and innovative consortium comprised of 13 senior investigators from 10 different institutions. It's an alliance of laboratories working collaboratively and interactively toward the mission of the HRP, the mission being to develop methods for replacing missing or dysfunctional cells in the inner ear. That is, the mission is restoring lost hearing and balance function in humans. It's a coordinated multi-center effort to hasten progress, to recruit more investigators, to provide new ideas and new tools to the entire field, not just ourselves, and to provide stable funding to a group of top contributors. Here are the members of this consortium that have already started. These are the founding members, and you can see they're from a lot of different institutions, and these are, I feel, the best investigators in the world in this area. The consortium members have met to develop a strategic plan to identify the next experiments and projects that will advance the field. The consortium members have identified how we can work together to expedite the research and best utilize each other's facilities and each other's skills. And the first HRP-funded projects will commence in early 2012, we hope. The first projects involve understanding the genes that control regeneration in birds and fish, and understanding how those genes are altered to stimulate regeneration, to identify specific cells that create the regenerated hair cells, to compare the genes that are turned on or off during regeneration, to alter the expression or function of those genes in non-mammalian vertebrates and see if they are critical for the regeneration process, and to test those candidate genes in mammals. Here are a couple diagrams of phase 1. We look at all the other animals that do regenerate hair cells, and we look at what genes are being expressed during that process. Okay, we track the cells and find out which exact cells are making each gene and what those cells become in the damaged epithelium. And then we take those genes and singly or in combinations, as happened in the animals that do regeneration, and we try and experiment them in different orders and in different amounts and with different cofactors in mammals. So, hair cell regeneration is now generally recognized as a valid research approach toward the eventual cure for hearing and balance disorders. Emerging understanding of the cellular pathways underlying cell cycle control and cell differentiation in the inner ear are yielding exciting opportunities for discovering therapeutic targets. The question is not if we will regenerate hair cells in humans, it's when. And we think the Hearing Restoration Project can make decades turn into years. Thank you very much. Yeah. I'm happy to answer any questions. The front row here.
Yes. The regeneration in the chicken's hair cells is quite fascinating and quite amazing, actually, that it comes back as much as it does physiologically. I was wondering if there have been any tests of frequency response to see whether or not the chickens are actually hearing any differently.
Yes. There's a number of studies of that in chick, and we also did studies of that in starlings and in finches. And the frequency response comes back almost exactly, except for the highest frequencies. The highest frequencies still show a slight hearing loss, but they start out the highest frequencies like with a 60 to 80 dB hearing loss, and they end up with a 20 to 25 dB hearing. Well, it depends on if you're talking about a songbird or you're talking about a chicken. A chicken, a high frequency is on the order of 4 kilohertz. A songbird, the high frequency is on the order of 6 kilohertz, 6 to 8 kilohertz. And they show the same pattern in terms of the loss of the higher frequencies for them. I think it's not a problem of the regeneration. I think of the hair cells, it's a problem of that they don't develop all of the right ion channel properties for responding to very high frequencies. That's my hypothesis. Yes, ma'am. We've also done temporal studies, by the way.
At this point, how do you think that regeneration process would be? Is it a surgical implantation or some kind of injection? Do you foresee that?
Okay. My guess is that, and it's strictly a guess because we don't understand the biology of it. My guess, and my colleagues and I are working on the idea right now, although it could change. You got to be flexible in science. But my guess is that what we're going to do is stimulate the surviving cells to either reprogram them to make new hair cells or to start dividing again and make new hair cells. I think it's most likely that it'll be from a local infusion of some compound or compounds into the middle ear or into the inner ear. If you put compounds into the, and we've done this, into the middle ear with a slow release kind of mechanism, you can get them to infuse through the round window into the inner ear. Maybe it's going to take direct injections into the inner ear. We really don't know.
How does that work in the same way for a person with genetic hearing loss?
Wow, that's a great question. One answer is that we could suspect that it won't, because the new hair cells will still have the bad gene, so to speak. Okay, another answer is that maybe in some cases, like a lot of cases of genetic hearing loss, there's a slow onset. So maybe we could put cells in. But of course, if we knew what the gene is, we could do gene therapy at the same time, okay, and transform that and put in the normal copy of that gene with whatever transcription factors we put in to stimulate hair. So all bets are off, and science is marching forward in these fields very, very fast. But I think you should ask that same question to my molecular biology colleagues, which will be here in a second. Yes?
I have a question. Mine's more on the line of the relationship between the hair cells in the cochlea and that in the vestibular system. You mentioned with regard to birds, but I don't think many people think about the fact that, or is it true, that when you have sensorineural hearing loss that may be severe, we also have some kind of a balance problem. And as you get older, we have two things going on, the cerebellar area and the vestibular area.
Right.
Are we looking at that at all?
There are many people that are trying to replace neurons. Okay, there are many people, we know that there are trophic relationships between the ear and the neurons they innervate and things like that. So I think that the brain, as we know from cochlear implant research and other research, is a very, very plastic system. That is, it can learn with new signals to solve the problems quite readily. And so our research is focused on replacing hair cells, be they in the vestibular or the cochlear system or both. And I agree with you. With aging, often we get loss of hair cells in both. We really don't know how to study the loss of hair cells in the vestibular epithelium quite as precisely as in the hearing epithelium. Excuse me. But I think that will emerge. But I think we do have to repair both. My colleague George Gates thinks that the first ability to repair is going to come in the vestibular epithelium because it's, in a way, a more primitive epithelium. It's not as highly differentiated. And we've seen more sort of signs of regeneration in the vestibular system. Also, the vestibular system is almost perfectly conserved between non-mammalian vertebrates and mammals. In the cochlea, there are some fundamental differences that you've seen. Yes, sir.
Have there been any studies relating the presence of Parkinson's disease as an antecedent to hearing loss?
I don't know, is the short answer. George? No, there haven't been, apparently. No. Just a sec, she'll bring you a microphone.
Why is so little time devoted to tinnitus? All kinds of people claim that they have cures for it, and none of them seem to work. And tinnitus is a very difficult experience to live with.
Right.
What is the problem there?
So there are many theories, okay, and there are probably fairly many kinds of tinnitus or tinnitus. It's not that nobody's devoted to it. Every drug company in the world would love to have a pill that they could sell for tinnitus, and as you know, most of them don't work. My thinking is that most tinnitus, okay, the perception is in the brain, but the pathology starts in the ear, and it's usually the tinnitus is in spectral, in frequency properties surrounding the area where there's hearing loss, the frequencies where there's hearing loss, indicating that those hair cells are probably weakened, okay, and they're not functioning as well, and they're probably releasing transmitter in an abnormal way. And then the brain also gets confused about that, and the brain hears these these different sounds when they're not there. I think if we prevent hair cell loss, we're going to prevent a lot of tinnitus or tinnitus. But it's not the case that nobody's working on it. Lots of people are working on it, but it's, again, not a simple problem because we don't know where its origin is, except that we know that people hear it.
One more question.
Thank you. Yeah.
Thank you. I certainly commend you and your colleagues on all the work you're doing. It's tremendous. I wondered whether any focus had been given to single-cell regeneration through low-level laser therapy, such as is being practiced in Germany. I happen to have been over there in regard to tinnitus I've had for 3 years, and it has been helpful. Perhaps not as helpful as where you're headed ultimately, but it certainly does help to improve hearing, and tinnitus, as the lady was just asking about, somewhat also. Having said that, one treatment doesn't work for everybody, as you'll know better than I do, but I just appreciate any comments you might have which could be helpful to other people in this condition.
Well, I don't know what the single-cell laser treatment is. It's kind of hard for me to imagine a single-cell laser treatment in the inner ear. But laser treatment, there have been a number of studies on the actual destruction of inner ear tissue, relieving tinnitus or tinnitus, and some people actually get their hearing nerve cut to actually relieve them of tinnitus. That's sacrificing your hearing because of that, and tinnitus can be sufficiently bothering that people want to do that. I don't know the single-cell laser treatment. That's what I've heard. Dr. Gates says if you cut the nerve, eventually it gets worse. It's sort of like referred pain, I think. But you're talking about something else. You're talking about it's stimulating the nerve. Well, you can stimulate the production of ATP by low-level stimulation. Is it an ongoing stimulation or one treatment, or what is it? Yeah, are there animal studies to know exactly what that's doing to the cells? Stefan, Andy, can you help me with that? No? He's finding it. I'm sorry, I can't help you with it. I'll look at it, okay, and see. I don't know of any animal studies. There are lots of animal studies going on in tinnitus, but doing studies in animals in tinnitus is pretty hard. It's like doing studies in animals in pain, because tinnitus is a perceptual problem, and asking an animal when it perceives. But people have developed behavioral tests for it. Maybe some of the best people are at Southern Illinois University and University of Buffalo.
Thank you very much.
Thank you.